Welcome to Lecture 32, in which I will discuss the molecular events that are responsible for training adaptations. In our previous lectures, we've talked about the numerous biochemical changes that occur in skeletal muscle as a result of several weeks or even months of endurance training. And we've also talked about how these changes are initiated from some sort of exercise stimulus. But what is that stimulus? So in this lecture, we're going to dive down deep into the muscle fiber and see what events are actually going on that causes these changes to occur. So I'll do something a little bit different on this lecture, and I'll put our learning objectives right here at the front. So our pages are going to be 305 to 310, and so some of the issues we're going to look at, what is the big picture of training-induced adaptations? I'll talk about that first. And then we'll get into the details about mRNA expression. What are the signals that cause a fast to slow fiber type shift? Which signals cause an increase in mitochondrial synthesis, also called mitochondrial biogenesis? And we'll look at the time course over which these molecular events occur. Let's begin with the big picture of training adaptations. Ultimately, the adaptations that occur due to exercise training, whether it's endurance training or strength training, are going to be because of an increase in some specific proteins within the muscle fiber. The process by which these new proteins are created is going to go something like this. You'll have some sort of a training stimulus right here at the top, and this is going to be a flow chart. And that will activate some set of intracellular signals that we will talk about. And then you will have the activation of some specific genes within the DNA. Of course, from that, we will create mRNA in the nucleus, and then the mRNA molecules will leave the nucleus, go out to the cytoplasm, and through the ribosomes, we will synthesize new protein. And that pathway, this flow chart, will occur with every single exercise bout. And so you'll form some stimulus in the muscle cell, you'll activate certain genes, you create protein. Now, a single exercise bout won't be enough to create very much protein. But if you do it day after day after day, ultimately the amount of protein will increase in the muscle fiber. The physiology behind this idea can be shown in figure 13.8, where on the left part we have the response of mRNA within the muscle. So it's the mRNA levels on the y-axis increasing over time. Notice that it's a 24-hour time period, and this is hours after exercise. The graph on the right shows weeks of exercise training on the x-axis, and then your y-axis would be your protein levels that are increasing over time. Let's think about how studies like this are done. So first off, you'll have a group of subjects. You'll have them do some sort of an exercise bout. And then, before the exercise bout started, the researchers will have taken a muscle biopsy. And then you can see on the left chart here, the left diagram, hours after the exercise, they will have taken on each of those time points another muscle biopsy from a muscle that was exercised. So if it's, say, a cycling exercise or even running, you probably would use the vastus lateralis. And so at 2 hours, and again at 4, 6, 8, 12, and 24 hours after the exercise bout, researchers would take a muscle biopsy and then examine it for the levels of mRNA for specific proteins that we're interested in, or I should say specific genes that we're interested in, that we think were probably turned on by the exercise itself. And so if we take an example, look at mRNA for protein 1, this could be something like, myosin, or actin, or SDH, succinate dehydrogenase, or lactate dehydrogenase, or maybe a glucose transporter like GLUT4. So if we look at the red line, mRNA of protein 1 at 2 hours, it is 100% elevated above the resting level. And then it keeps going up for a full 8 hours after the exercise bout. And then it starts to come back down. Turns out that RNA, or mRNA, actually isn't very stable in the muscle. And after it's no longer being produced by the nucleus, the levels will fall pretty rapidly. Conversely, if we take the green line, the mRNA of protein 3, we could see it's only about 25% increased at 2 hours. At 6 hours, it peaks at about 200% and then rapidly declines. And even by 12 hours, it's almost back to normal. But notice that all three of the proteins are back to the baseline levels by 24 hours. If we look at the overall response of these mRNAs, it looks like protein 1, the mRNA for protein 1 and the mRNA for protein 2 had fairly similar responses. Protein 1 peaked a little later than protein 2. But if we just take protein 1 and we color this red, we can take an area under the curve, so to speak, and look at all of this area here is what was affected by or underneath the mRNA of protein 1. So that's the total response, sort of a concentration over time. Now if I change it to green and we can look at just the mRNA response for protein 3, and that would only be this section underneath the green line. And you can see if you quantified the area, let me start over with that and make it clearer. Now we can just do the green. If we quantified the area for just the green area under the curve, it'd be much less than for the red area under the curve. So that just means that there is a less response or less activation of that gene after this exercise bout. So we just saw the mRNA response for one day or one exercise bout. And now, keep in mind, if someone's training day after day after day, that change, that increase in mRNA and then back down to the resting level will occur day after day after day. And when mRNA levels are high, that means there's lots of copies of that mRNA in the cytoplasm. And so the ribosomes can be making lots of copies of the protein. So now if we go over to the diagram on the right, it shows instead of days of training, now we've got weeks of exercise training, starting at 1 week and then going up to 12 weeks. And so what we're seeing is an accrual of those protein levels week after week. And notice that the red protein, that is protein 1, it increases by the largest percentage. So that notice that the y-axis here is percent above the untrained value. So that by 12 weeks, we have an increase in about 130%. Now the green protein, which remember had the smallest response of its mRNA, it shows the least increase. And so it gets to about, after 12 weeks, maybe 100% increase. That's about a doubling of the protein in the muscle fiber. So that sort of sums up what's going on inside the muscle fiber. We do an exercise bout. We've activated some stimulus. We will produce some mRNA of a specific gene. And then the cell will create more protein over weeks and months. And that is what constitutes then the adaptation. So now the muscle fiber is better able to handle this challenge to homeostasis that is being presented by the exercise bout. If this were a strength training protocol, we could assume that, say, the red protein might be myosin, and the blue one is actin, and the green one is, I don't know, troponin or something like this, right? And those all would then increase as the muscle fiber started producing more of these contractile proteins. Then, of course, what would happen? The muscle would get bigger as the amount of these proteins would increase inside the fiber. So now that we know what happens inside the muscle fiber, the next question, of course, would be, well, how does this happen? How do the signals arrive at the nucleus, or how are they even created in the first place? But before we get to that, I want to talk about why this happens. There is one theory that suggests that these proteins, whether it's actin or myosin or a transporter, they are increased because the cell is preparing to handle the next time this exercise bout would occur. I don't think that's actually the case because, of course, the muscle itself has no idea that another exercise bout is coming. The cells can't anticipate anything is going to happen to them. They can only respond to their environment. And so the environment we're creating by exercising, of course, is a homeostatic challenge that we have programmed. There are programs in the cell, that is the signaling pathways, that will then activate the genes. Now, the increase in the amount of these proteins, of course, will help whenever a subsequent exercise bout is is encountered, but the response can't be because there might be another exercise bout. It is simply because we've activated these signaling pathways that then result in gene activation and protein synthesis. So now let's go ahead and look at the signaling pathways involved in this response that is leading to these increases in proteins. The signals that become activated during exercise, we can divide into both what we call primary pathways and secondary pathways, as shown on this slide. For the primary pathways, these are essentially the events that are happening in the muscle, or sort of the main signal that is creating the response. The first one is mechanical stimuli. That's things like stretch and tension on the muscle. That's primarily going to be involved in the response to strength training. The other three, increase in calcium, free radicals, and phosphate energy levels, are going to be active in endurance training responses. Calcium is a large signal. It makes sense because with every muscle contraction, we have to liberate calcium from the SR, which then activates muscle contraction. Well, it can also stimulate specific signaling molecules. And so this pattern of increased calcium, contraction after contraction after contraction for a long period of time, will create some certain pattern of response. We'll take a look at how that works. For free radicals, we know that with endurance training, there's an increase in free radicals, mostly because of mitochondrial respiration. And that also will then activate a certain signaling pathway. The third one is phosphate and energy levels. There are sensors within the cell that respond to changes in the ratio of AMP to ATP. So, of course, the higher your metabolic rate, the faster you're breaking down ATP to ADP. Then we have another enzyme, myokinase, that can take two ADPs, transfer a phosphate to make another ATP, but then you're left with, of course, an AMP. So AMP levels, adenosine monophosphate, that will increase with exercise. And there are proteins that will sense what we call the energy charge of the muscle, and that's that ratio of AMP to ATP. So those are the primary pathways. We'll take a look at how those activate the secondary pathways. The secondary pathways are, now we're talking about specific molecules that involved. These are proteins, and they become activated mostly by phosphorylation. So we're dealing with lots of kinases. Kinases are enzymes that add phosphates to other groups, other proteins, or it could be other target molecules, but usually proteins. And so these proteins then will be activated by any number of the primary pathways, and then they will set into effect, ultimately, transcription of a specific gene. To discuss the roles of each of these secondary pathways and these specific molecules, let's jump ahead to figure 13.9, which shows the actual pathways themselves and then the placement of each molecule within it. I should point out this is for endurance exercise-induced adaptations, and so these are the signals that are going to be involved, that are be activated by endurance training. At the top of the figure, we have our stimulus, which is endurance training. And then right below that, we can see our primary signals: calcium, AMP to ATP ratio, and free radicals. And the up arrows indicate that those are all increasing. So here's our primary signals. So let's take each of these primary signals and see what they do to the secondary molecules. So let's start with calcium. And we can see, if we follow the arrows, that an increase in calcium is going to activate two of these secondary molecules, calcineurin and CAMK. So calcineurin is a phosphatase. That means it removes a phosphate group rather than adds one like a kinase does. But it activates downstream molecules, and it is involved in the fast to slow fiber type transition that occurs with endurance training. CAMK is calmodulin-dependent kinase, and it's activated by calcium, of course, and when it binds calcium, it then helps to activate what is, as you can see, sort of the linchpin molecule here of the secondary signals, and that's PGC1 alpha. We'll always use the abbreviation here because the full name is peroxisome proliferator activated receptor gamma coactivator 1 alpha. This is a activator of gene transcription, obviously in the nucleus, and it's the activator, the main activator for mitochondrial biogenesis. When I was in graduate school, both PGC1 alpha and ampk were discovered, and those were enormous, humongous, big breakthroughs that generated lots of attention in the metabolism field, and for good reason. You can see that PGC1 alpha is also going to cause or contribute to the fast to slow fiber type shift. Our second primary signal is the AMP to ATP ratio. We can see that it will activate AMPK. This is adenosine monophosphate activated protein kinase, or what we call AMP kinase. And so as AMP levels go up, this protein gets activated, and then it also helps activate PGC1 alpha, which then will contribute to more mitochondrial biogenesis and this fast to slow shift in fiber types. The third primary signal here is free radicals. I'm not a big free radical fan, maybe because I don't know much about them, but one of the authors of our textbook does a lot of research on this, so he includes it in a lot of the stuff that he does. That's Scott Powers. But we do know endurance training increases the free radical production in the cell, and that's going to activate two of these signaling molecules, P38 and NF-kappa B. The full name of NF-kappa B is nuclear factor kappa B, and so it is a transcriptional activator and that is active in the nucleus, and where it's how it gets its name. But these two molecules, well, P38 will activate PGC1 alpha, and NF-kappa B will activate synthesis of antioxidant enzymes to deal with those free radicals. And again, I won't pay much attention to the free radical story, or part of this story, I guess. I'll concentrate mostly on the fiber type transition and mitochondrial biogenesis, which are going to mainly work their way through PGC1 alpha. In fact, I'll double down on my interest in that and show you a results from an article that came out, I think this was maybe in the late 90s. I remember reading this when I was in graduate school. And the main point here is, as it shows at the top, activation of AMPK by ACAR. This is a drug that mimics AMP. It's a lot like AMP, but it can easily get into the muscle cells. And this increases aerobic enzyme activity in skeletal muscle. So what we have are three charts here. The one on the upper left shows an increase in malate dehydrogenase. The one on the bottom left is succinate dehydrogenase. That's one that we're familiar with. And the one on the right is citrate synthase. So these are all Krebs cycle enzymes. And so the researchers just gave these rats, the group of rats, this drug, ICAR, and they just would inject it into them. And then after several weeks, they noticed that AMPK levels would increase in white muscles and soleus muscles of the rats, which is red, primarily. Well, it's slow twitch red muscle, so it's primarily type 1. The white muscles will be type 2X, and then the ones that are just labeled red, those would be fast twitch red. Those would just be your type 2A muscles. So for some reason, the white muscles and the soleus muscles for all three of these enzymes showed an increase in AMPK. So this was indicating that if you activated AMPK, which of course it would be like in the cell, what's happening is that AMP is going up, then you'll see an increase in this enzyme activity, which is indicative of greater oxidative capacity by the mitochondria. So they were inducing this reaction without even exercising the rats. These were just sedentary rats. Finally, let's take a look at the time course over which these responses will occur. If you look at this figure 1310, it has this stopwatch figure where we've divided it here into then what happens over seconds, minutes, and then hours. So in the first few seconds, of course, right at the onset of exercise, you'll have an increase in those primary signals, an increase in calcium, increase in free radicals, and an increase in AMP. And then over the next several minutes, we'll start activating the secondary pathways and those molecules involved, calcineurin, calmodulin kinase, ampk, P38, and F kappa B. And then, of course, over hours, you'll see an increase in PGC1 alpha and the onset then of mitochondrial biogenesis that will continue day after day after day, week after week. So there's an overview of the molecular events that ultimately cause the adaptations to endurance exercise training. When it comes to strength training, I'll address those in a later lecture.